today we will continue with our journey of reproductive biology of angiosperms, and what we are going to discuss today is the endosperm. So far we have been talking about the reproduction in angiosperms, and you would recall that there is a phenomenon called as the double fertilization in angiosperms. Now this double fertilization is because there are two male gametes which are present, and one of these fuses with the egg cell while the other fuses with the polar nuclei present in the central cell. Because these two incidences of fusion occur, it is called as the double fertilization. You would recall that this fusion of one male gamete with the nucleus of the egg cell or the female gamete is what leads to the formation of a zygote and subsequently formation of embryo, and this process of fusion is called as syngamy. The second act of fusion is the triple fusion, where the second male gamete fuses with the two polar nuclei which are present in the central cell. So, there are basically three cells which are three nuclei which are fusing, one of the male gamete and two of the polar nuclei, creating a triploid structure. And this is what we call as the primary endosperm nucleus. This is the product of triple fusion resulting in a triploid nucleus. So what we have learnt is that syngamy leads to the formation of zygote and embryo, and this primary endosperm nucleus leads to the formation of endosperm. So double fertilization leads to formation of both the embryo as well as the endosperm. Syngamy leads to formation of embryo and the triple fusion leads to the formation of endosperm. Formation of the primary endosperm nucleus is always preceded by several ultrastructural changes in the central cell. Primary endosperm nucleus follows different developmental pathways and it can form different types of endosperm. Now this picture is a repetition of what we have already studied during the lecture on fertilization. You can see that in figure A, it is depicting the mature embryo sac. There are three antipodals, there are two synergids, one egg cell, central cell with a vacuole in the center, and there are two polar nuclei. So this is actually an embryo sac before the arrival of the male gametes, the one on the extreme left, that is A1. In B to C, you can see that the two polar nuclei which are present in the central cell, they move close to each other, and soon after the membranes of these polar nuclei, they fuse. The last picture, or the D1, you see that embryo sac has secondary nucleus, that is the polar nuclei, the two polar nuclei which are present in the central cell, they have fused in this picture, okay. And this is also happening at the same time when the pollen tube is entering the synergid. If you remember, this pollen tube is the carrier of the two male gametes. The pollen tube enters one of the synergids, which we call as the degenerating synergid, and this is where it liberates its contents by bursting. So in one of the synergids, both the male gametes are liberated, and from here one male gamete moves to the egg cell and the other moves to the central cell, right. And so one male gamete fuses with the egg cell, that is what we said is syngamy. It leads to the formation of zygote and then embryo. The other male gamete from the synergid, it moves to the central cell and then it fuses with the secondary nucleus which has been formed by the fusion of two polar nuclei, creating a triploid structure which will give rise to the endosperm formation. So Just repeating what we have said, the two polar nuclei, they fuse together and form a secondary nucleus before fertilization. The nuclear membranes here of the two nuclei, they evaginate, they make contact with each other and eventually they will fuse. The secondary nucleus occupies the micropylar portion of the central cell, so that it remains close to the egg apparatus, and a big vacuole is present towards the chalazal end of the central cell. The central cell becomes densely cytoplasmic and it has a higher number of cell organelles, and this high number of cell organelles indicates that the central cell is metabolically very active. During fertilization, the secondary nucleus, it fuses with the sperm nucleus, giving rise to triploid primary endosperm nucleus, and we abbreviate this as PEN. The fertilized central cell is called as the primary endosperm cell or the PEC, and the PEN or the primary endosperm nucleus is confined to the primary endosperm cell, which is also the central cell of the embryo sac. Now, what is an endosperm? We have been talking about endosperm. So, if I have to define, endosperm is a tissue which is produced after fertilization inside the seeds. That is why it is called as the endo, that is inside sperm seed of most of the flowering plants. It surrounds and it nourishes the embryo in the seeds of the flowering plants. So, it is very important that the endosperm is present when the embryo is growing, because endosperm plays a key role in the growth and development of the embryo. In most angiosperms, this endosperm is short lived, and because when the embryos are growing, the developing embryos, they consume the endosperm completely before germination. As we have discussed, the endosperm is a triploid structure. This triploid structure provides nutrition in the form of starch or oils or proteins. Okay. So, if you imagine a grain, you all have seen grains or seeds or kernels, they are made up of three parts. One part is the bran, second is the germ or the embryo, and the third is the endosperm. Now, what is bran? You would have heard your mother saying that bran is important for our gut, because bran is the multi-layered outer skin of the edible kernel. It contains important antioxidants, B vitamins and fiber. Because of the fiber which is present in bran, bran is a good source of nourishment for our system. Germ is the embryo which sprouts into a new plant. It contains many vitamins, proteins, minerals and healthy fats. Especially it is rich in B vitamins. And then comes the endosperm. Now, endosperm is the food of the germ, or it is the germ's food supply which provides energy to the seed to germinate. By far, this endosperm is the largest portion of the kernel, which contains starchy carbohydrates, proteins, and also it has small amounts of vitamins and minerals. So, what you see here is, now this is a picture which shows you one grain, right. It is divided into three portions like I was just discussing with you. Endosperm occupies the largest portion of this grain. Endosperm is rich in carbohydrates, proteins and B vitamins. Okay. Endosperm is the one which is nourishing the germ. Germ is also called as the embryo. It also has different vitamins. It is rich in minerals, phytochemicals and antioxidants, and this portion of the grain is going to sprout into a new plant. Both the germ and the endosperm here, they are covered by a layer of bran which is rich in fibers, vitamins, proteins and minerals. So there are two types of seeds. You would recall that there are endospermous and non-endospermous seeds. Broadly speaking, endospermous seeds are those which contain endosperm, and non-endospermous or the ex-albuminous seeds are the ones where there is no endosperm. Now, why does it happen that some seeds have endosperm present in them, whereas the others, they do not have endosperm present in them? In non-endospermous seeds, of course, endosperm is present in the initial stages. The endosperm here, it's completely absorbed at the time of maturity. So, if we observe a seed which is mature, there is no endosperm like in peas, beans and cucurbita. Why? Because the growing embryo and the cotyledons, they have consumed all the endosperm. The endosperm is completely absorbed, and the fleshy food storing cotyledons, they nourish the embryo as the embryo germinates. Then come the second type of seeds, which are the endospermous or the albuminous seeds. The endosperm here persists till germination of seed or when the seedling is formed. Mostly this is seen in cereals, it is also seen in coconut and castor. Now, what is happening here is the cotyledons are thin and membranous. They serve to absorb stored food from endosperm upon germination, right. So, endosperm here plays a major role in the growth and development of the seeds. Endospermous seeds or the seeds which contain the endosperm are also called as the albuminous seeds or the endospermous seeds. Endosperm, because is the one which provides nourishment to the growing embryo, endosperm is responsible for the economic importance of all cereal grains and also oil seeds. You all would have eaten coconut. Now, coconut is a very important example where the endosperm is very prevalent. Okay. So, coconut is one example of being an endosperm. Endosperm, I told you in the beginning that endosperm is a tissue on which the embryo is dependent, but depending on the development and on the differentiation, there are different types of endosperms, and we can also classify this endosperm on the basis of cell wall formation. Of course, when cell division takes place, there are many nuclei which are formed. Now, the defining character here is the formation of cell. If there is cell wall formation immediately when the nucleus divides, then the appearance would be cellular. But if there is no cell wall formation, but the nuclei they keep dividing, then it gives a multinucleate appearance. So, on the basis of cell wall formation, we have three types of endosperm. The first one is the nuclear endosperm formation. You can also see in this figure that one primary endosperm nucleus, it undergoes one mitotic division and there are two nuclei which are formed, right. On the top of the diagram is the embryo. Okay. So, these two nuclei eventually will undergo more mitotic divisions and there will be several nuclei which are present in this cell, that is there are free nuclei which are present. Now, repeated nuclear divisions they take place, and if at all a cell wall is formed, like you see in the last diagram. This cell wall is formed after free nuclear divisions, and that too at a much later stage during development. So basically the endosperm has many nuclei which are present in a cell, and these nuclei during early stages of development are not partitioned by cell wall formation. Then the second type is the cellular endosperm formation. As the name suggests that the endosperm formation is cellular. Cell wall formation here is coincident with nuclear divisions. You can see here that this primary endosperm nucleus, it divides into two, and immediately as two nuclei are formed, there is also formation of a wall which separates the cell into two portions. Okay. Cell wall formation is coincident with nuclear divisions. So, subsequently when there will be more divisions which will be taking place in the nuclei, each nuclear division is followed by cell wall formation. So, the endosperm gets a cellular appearance, right. So, you can see that the last picture or the diagram here, it gives a very cellular appearance. There are no free nuclei present here. So, such kind of endosperm is called as the cellular endosperm. The third type of endosperm is the halobial endosperm formation. Okay. Now, this halobial endosperm formation is a combination of the first two types of endosperms we have just discussed. We have talked about the nuclear endosperm and the cellular endosperm. The first one, nuclear endosperm, there is no cell wall formation till the last stages of development, whereas in the cellular endosperm, each nuclear division is accompanied by cell wall formation. Now, what you see here in this diagram is that there is primary endosperm nucleus which is present towards the bottom portion of the cell, and this primary endosperm nucleus divides into two. Okay, two nuclei are formed, and this nuclear this division of the nuclei is immediately followed by a wall formation. So, now we have two cells with one nucleus each. The upper chamber after cell division is larger than the lower chamber. Upper or the micropylar chamber has more number of nuclei, because here the number of divisions, you can see the number of divisions, nuclear divisions are not followed by wall formation. So, what you see is that there are a large number of nuclei lying freely in this micropylar chamber. The lower portion of this cell is smaller, that is the chalazal chamber, and here each nuclear division is followed by cell wall formation. So, the upper chamber or the micropylar chamber has free nuclei present, the lower chamber has cell wall formation, and you can see that it is a cellular structure. The last or the final stage of development of halobial endosperm, which you see here, is that even in the micropylar chamber there is cellularization at the periphery which has happened. The rest of the nuclei lie freely in the chamber. So, what we understand here is that the first nuclear division is followed by cell wall formation. Later, there are two portions formed. One will exhibit the free nuclear kind of endosperm, and the other portion exhibits cellular endosperm. So, halobial endosperm formation is a combination. The definition says a cell wall is laid down between the first two nuclei, after which one half develops endosperm along the cellular pattern and the other half along the nuclear pattern. But the picture which you are seeing here, it's actually the cell has not been divided half and half. The micropylar chamber is slightly bigger than the callus. Such kind of endosperm formation is characteristic of monocots. Now, we will talk about each type of endosperm separately. Nuclear endosperm is the most common type of endosperm development. Once the fertilization is accomplished, the primary endosperm nucleus, it divides repeatedly, that is 1, 2, 3, 4 multiple divisions they occur, and each of these nuclear divisions are not accompanied by cell wall formation. So, what does it result in? If there are many nuclei which are present in a common cell, then this is a cenocytic stage, and this formation of a cenocytic stage is characteristic of nuclear endosperm. So, numerous nuclei, they remain suspended in the primary endosperm cell or the central cell of the embryo sac, and this stage is called as the syncytial phase. You can see here in the diagram that the primary endosperm nucleus, it divides. First mitotic division, you can see that there are two nuclei which are present, and then from 2 there are 4, then 8 and 16 and 32. There are multiple nuclei which can be seen in figure C here, okay, and there is a small vacuole which is present in the center. Eventually, this vacuole, it enlarges and it pushes the nuclei towards the periphery. And then at a much later stage, that there is cellularization of this kind of endosperm also, but not the entire endosperm becomes cellular. And the most characteristic feature of such endosperm is the cenocytic stage or the syncytial phase, where there are multiple nuclei which are present in the central cell. Syncytial stage is followed by cellularization, which occurs after a specific number of divisions. Now, this number of divisions, it varies from species to species, and it is characteristic of a particular species. Like in Arabidopsis, this cellularization of the endosperm, it starts after 8 rounds of division. Okay. To begin cellularization, like I showed you in the previous picture, a vacuole in the primary endosperm cell, it enlarges and it pushes the nuclei towards the periphery of the cell. You can see here that initially the vacuole is very small and eventually it grows in size, and at a later stage it occupies the major portion of the cell, pushing all the nuclei towards the periphery. Okay. So, the cell walls then they get laid down in a centripetal manner. Centripetal manner, you would remember, is the one which begins from the periphery and moves towards the center. The cell walls, they get laid down in a centripetal manner. This initiates in the micropylar region. Once cellularization is complete, that is all cells have been formed, the peripheral cells of the endosperm, they undergo synchronous cell divisions to form 5 to 6 layers of cells. Now, such kind of endosperm is seen in our cereals, wheat, rice and maize. Okay. We will continue the same in the next lecture. Thank you.